Hello, my name is Aneequa and welcome to Advanced Session 1 for PhysiCell Workshop 2023. And in this advanced session, I would be talking about boundary conditions, custom variables, user parameters, and C++ searches. So, uh, the purpose of this session is to introduce you to work that can only be done in C++. As you have seen in the previous sessions, that most of the things that a basic model needs can be achieved using PhysiCell Studio and rule-based languages. But still, each particular project, each particular model has its own requirements that are not achievable using the default parameters or just changing the parameters that are available in the studio. So if you want to do some customization into your model, you will have to go to C++ code and add your stuff there. So this first session is just to introduction where you will go for C++ code, what are the things that you can access in C++, and then you can later in the next sessions, you would go and see the examples of working with C++ code. So I will go through these things, for example, how to access user parameters in C++, um, how to define some custom cell variables, how to query for cell definitions inside your code, how to query for the microenvironment, how to see boundary conditions, and how to access the dictionaries for signals and behaviors that you have seen in the session 1 and 2. So where would you go and edit these things in C++ code? For a normal basic project, once you load the project, there is a custom CPP and custom.h file that are in the custom modules folder. And you will go there, and for the most part, you will be editing custom.cpp and custom.h. And for some cases, you can go to XML file and you can edit there. But most of the things that you can edit in XML file that can be edited through Studio. So for C++ function, custom.cpp is the main file here, where you will be adding your own custom variables, custom functions, and different customizations for the code. So first are user parameters. User parameters are the one that you add in the PhysiCell Studio. That's how we recommend it. You go to the user parameters section, you add parameters there, or you can add them in XML manually, where you will go to XML file. And here I'm showing a synapse of it, where you go to the user parameter section in the XML file, and you can add a new parameter there. And how will you access these parameters in your C++ file? This will be you can do parameter doubles onc_protein main. So this will be accessing this one, which is onc_protein main double, which the value is 1. So through this statement, you can access these variables. So ints, bools, everything, you can access them in your C++ file, and then you can use these values to do whatever you want to do with those values. Okay, next important thing is custom variables. Custom variables are so each cell has some data attached to it, and custom variables are the things that we attach to each cell. Again, these are cell level things. The user parameters are the overall simulation level things where you can access them, but custom variables are we attached to each cell, and we can add these custom variables using PhysiCell Studio. You have seen that. But if you want to add them in C++, although you don't need to add them now while you have availability of custom PhysiCell Studio, but suppose you just want to go to C++ code and add the new variables there. Maybe you love C++ much more. So you can go to cell defaults custom data add variable, and you will give the name of the variable, dimensions of the variable. In this case, I gave the dimensionless, and the initial value. So it will create a new variable for cell defaults whose name would be energy, and its default value will be 0.5. Similarly, you can get the value from the parameter. So suppose you want to add the variable energy, which is dimensionless, but the value that you are initializing is you are getting it from the XML file. So you will get parameter.double cell default initial energy. So the cell default initial energy will be a parameter in the user parameters, and you can get it from there and initialize your custom custom variable with that value from the XML. And once you have this value, how will you access it? So this is now each cell default cell has this variable attached to it, which whose name is energy. You can add alpha, anything else, and its value is 0.5 or whatever in the user parameter is, depending on which statement that you use. And after that, what you will do is you will have whenever you need to access it, first you need to access the index of that variable. So you will go to the PCell. PCell is a pointer to the cell. You will get custom data that find variable index energy. So custom data is all the custom uh, custom variables that are attached with that cell. And from there, you will find the index of energy and you will save that index. And then from that custom data vector, you will give the index of this particular energy, in this case, a variable, and you can access it to the P cell. So P cell custom data variable, and that will give you the actual value of whatever the value of energy is in that. And you can change it, you can modify it in your custom functions that we'll be discussing in the next section. Okay. Then once I said that you can add the cell defaults to it, then there are different cell definitions that are there. So if you want to access a particular cell definition, for example, a blood vessel or a tumor cell or an immune cell, so how will you access that? So either you know their index, uh, which is available. You can check it in the studio, which indexes each cell type. You can check it in the studio. You can go to XML file, and each cell type where it is defined, it has an index, and you will go to the vector of cell definition by index and give it that particular index, and it will return you the pointer to that particular cell definition. But if you don't want to do that, you don't want to check which index it is, you can still search it by human readable name. So for example, if you want to check what is the cell, I want to access the cell definition of blood vessel. So you can give the blood vessel and find cell definition. This function can be really helpful to search for that particular cell definition, and it will return that cell definition, and it will be, again, it will be a pointer, and then you can use this to modify it, or you just query what are the different values of the cell definition. Okay, so these were the cells. Now going towards another thing that we can access in C++ is boundary conditions in microenvironment. So we know that microenvironment is the background diffusion processor, that is where the different chemicals are diffusing. And important thing to notice is that each cell, uh, so PhysiCell is not lattice-based. So there can be multiple cells in one voxel, but the diffusion solver happens on a mesh. So there's a background mesh where diffusion solver is acting, and each unit of the mesh is a voxel. So one cell, in one voxel, there can be multiple cells, and we want to see where in the environment that particular cell is, we will get the pointer to the cell and we'll get the current voxel index. So we'll see which voxel this is. And then based on that voxel index, we can also say that microenvironment find density index. So density index will give you the index of that particular substrate. So this will give you where the position of the cell is, and this is applying on microenvironment will give you what is the density index of particular resource. For example, here it is resource. It can be oxygen, it can be glucose, it can be some drug that is added to the microenvironment. And if there is nothing diffusing in the background, there is no substrate in the environment, the function will return -1. And so be careful of that. So once we have the index of the resource, we can see that what is the value of that resource for particular cell. So one cell is in a voxel, and it wants to see what is the value of resource in that voxel, because that's the value it will see. And so it will get the nearest density vector, and then it will give the index. So for example, resource is the index 2. So it will give the index number there, and then it will give you what is the value of resource there. If we will just access the nearest density vector, it will give you a vector of size n, where n is the total number of substrates in the microenvironment in that uh, particular simulation that you are working with, and you can access that, and it will give you the value of that particular substrate in that voxel. Okay. So what can the cell do? Cell can see the gradient of the index and the value of the index. So you will get the nearest gradient 2, so it will the gradient of the substrate number 2. Again, this is C++. Usually it's the third substrate, 0, 1, 2. We can access the mesh. We can access the voxels of the mesh. So if you want to see what are in all the variables, suppose you have a particular application where you want to add something in all the voxels at some point in the simulation. So if you want to add that, what we can do is we can iterate over all voxels in the mesh, and it will give you the you can iterate over them, and you can change something there, you can add something there. For example, at some point in the simulation, you want to add a drug to some voxels in the system, you can do that. Okay. Another important thing that we can do with microenvironment and without the boundary condition is that our Richelieu nodes, we cannot do them with the studio if you want to do it. So first, the concept of Richelieu node is that we can make one or multiple voxels inside the microenvironment which can act as source or sink. So again, these are not the boundary voxels which we can control by studio, and we can say maximum value at 1 and minimum value at 1. We are saying somewhere in the middle of the microenvironment we want to add Richelieu nodes. So we can add that. We can use add Richelieu node, and we will give you it a voxel index, which voxel we want to add the Richelieu node to, and what are the values of the vectors that you want to see. So this vector would be size n, where n is the number of substrates in the system. So if there are 3 substrates, it will get size 3, and it will set the values to that. So for example, I want to start a sink. So for the sink, I want everything to be 0 there. So this vector would be size 3, 0, 0, 0, and we can add Richelieu node and that particular voxel index, and we'll set the values to 0. And for that voxel, everything will go to 0, and it will start acting as sink for everything that's in the periphery, or it will affect eventually the whole microenvironment. So these kind of changes like add Richelieu nodes, these can be helpful. For example, you want to mimic some kind of vasculature, so you can add nodes into the microenvironment where nodes can act as vessels, and where certain level of oxygen is always maintained, and it is diffusing from there. So these kind of things can be achieved for using Richelieu nodes. Okay. So we did the microenvironment, we did the cells, and the other thing is signals and behavior dictionaries that we discussed a lot in the lectures today. But now I would say that I would explain how these dictionaries and signals can be accessed using C++. So as we talked about it, a dictionary of standard signals are there. So signals affects the cell phenotype of one particular cell. It detects the signals, and according to that signals, it behaves. It's an agent. We are doing agent-based modeling, so that's the basic structure anyways. So we have a cell, it gets the signals and it behaves. So we have a signals dictionary where all the signals that are available for that cell phenotype, it's there. And then we will have a behavior dictionary that what things can be changed. So what kind of signals The signals can be pressure, level of oxygen in the environment. It can be how many cells I am in contact with. These all things can be signals. Behaviors can be changed, the motility rate, change the proliferation rate, change the death rate. So if there is hypoxia, there is cell death. So this kind of behaviors can be controlled by getting the signal and controlling the behavior. And we do that usually now by the rule-based language, but sometimes you don't want to use the rule-based language, or in the rule-based language we're using the hill function and you want to use some other function for that. So you can access the signals and access the behaviors. So here again, I gave the example of some common signals. For example, extracellular, intercellular substrate concentration, substrate gradients, contact with dead cells, contacts with cell of type X, for example, macrophage. And if you get a contact with that, what will happen? Damage, pressure. And this function, if you write it in your C++, it will, once the system start running, it will give you display the signal dictionaries. And here, I think it's a previous version, but it's the heterogeneity signal dictionary. So basic default uh, default project that uploads starts with PhysiCell. That's heterogeneity. And if you display the signals for that, these are the signals that are available: oxygen, intracellular oxygen, oxygen gradient, pressure, volume, contact with cancer cells, and so on. So how will we access that signals in our C++ program? So we want to access these signals and want to change behavior according to that. So first we want to read these signals. So there are different ways to read the signals. You can get the index of a name signal. For example, signal name is oxygen or pressure or something else. So you can give the string name and human readable name, and it will give you the signal index if it's present. And how would you know it's present? Because you would know it by the dictionary list of dictionary that the signal is present. And or you want to get a vector of indices. It's not one index that it's not one signal that you are interested in. You are interested in a vector of signals. So you can get a vector of indices for a vector of named signals. Okay, or we can display the name of signal with the given index. For example, we know what is that signal index 0. Just display it and you can go from there. Also, we get a vector of all known signals. Again, this is here you are reading it in a string. Here you are using in a double vector, which is giving you the indexes. Get a vector of all cell contact associated signal for the cell. So get cell contact signals and it will give you that one. Get selected signals, pass it at a vector of indices, and it will return you a vector of those particular indices signals. So here you are getting the index of the signal, and here you're getting the value of the signals. For example, you will pass it indices 0, 1, and 5. It will give you value. What is the value of the signal 0? What is the value of signal 1? And what is the value of signal 5? Similarly, we can get those values by giving the name or the string of names. Sorry. And if you want to get single signal, you can give it an index or give it a name, and you can access it. So you can get single signal by providing it an index or providing it a name, which is a string. So in this way, you can get the signals from the inside your C++. So these functions, keep them handy if you want to use them. Most of the custom functions that you will be writing, you will get some signals and you will attach certain behavior to it. So you will read the signal, read the value, and according to that, according to your requirement, maybe there's an OR function, maybe there is maybe there's some other function that I can't imagine right now, which is not hill function, and you want to do that, or you want to do a particular version of hill function, you can do that. And then once you have read that signals using the functions that I have described below, you can use any of those functions according to your requirement. Now you want to use those readable values to change the behaviors. So here we have the behavior dictionary. So again, this is the by default heterogeneity sample, and it is the behaviors for that. You will might have different behaviors for your particular project, and you can change secretion target, uptake, exported. I think there would be a change in it in the newer version. I think I have the previous version of PhysiCell here, but mostly it's cell progression, attack rates, fusion rate, transformation rate, and you can display the behavior dictionary, and you can see what are the behaviors available for you to modify using um, using this. So how would you uh, access the behaviors? You can get the find behavior index. Again, the same thing. Um, get the index of the name behavior. You can get behavior indices in the form of a vector. So you can get one index or a vector of the index. You can get the string, whereas behavior name, a human readable name for that, or you can get empty behavior vector for that. You can set behaviors. Now, so for set behaviors, you will change the value of the behavior. Here you are getting the indexes, and on these ones, you are changing the behavior. So for example, you want to change the motility function, or you want to change the proliferation rate. So you would give the index for that, and then you will give the parameters. So if you'll give the parameters, you will it will change according to those according to normal Hill function. Um, you can change it the cell phenotype. You you can access it by name. You can access it by index. You can get a whole vector, or you can get a single behavior, and based on that, you can change the behavior. So you will give the cell and the signal name, and which parameters, how much value do you want to uh, tune up or tune down. So these are the functions for behaviors. And after that, I would go through some of the best practices that we recommend while working with C++, um, things to remember and the best practices. So in any customized cell function, you can access the microenvironment at its location. You cannot access that would be helpful for if you want to see where the cell is and what's the value of the microenvironment there is, and if you want to change any behavior based on that. We don't want you to hard code the index substrates because that would not be a good practice, because if somebody changes the substrates, or if you delete some substrates and all the index of the substrates are different, uh, that that can affect your code. So every time you want to access the substrate, get the substrate by the human readable name, and then work from there. So what you will do is you will search for the index of substrate and store the result in a static variables. And with the static variable, it will be only, it will not be accessing on every time in the loop. Um, Human readable names are better because again, they're human readable. You can remember them. You can change them. For example, stem cells, differentiated cells. You have vessels. You have bacteria. Whatever you are, use a human readable names that are easy to remember and that represent what your cell particularly represent. Again, dictionaries are the best way to add behaviors and signals. You can get other signals also which are not in the signal if you want to, but dictionary is a straightforward way you can get signal and get behavior functions that I discussed in the previous slides, and that will help you get to the exact behavior, and that will help you make your C++ code more cleaner instead of doing further searches. So that's it. Happy coding, and these are the funding acknowledgements for our side. And in the next session, you would see how a custom function is written in C++ for a PhysiCell uh, for a PhysiCell model. Thank you. Okay, sure.